Good morning, ladies and gentlemen. Enhanced tolerance to ion toxicity enhances grain yield in acid soil prone to flooding and terminal drought. This is the title of my talk, and I think this is the right venue to provide you with a summary of our suggested approach to tackle the widespread problem of constraint of a growing crop in acid soil prone to flooding, waterlogging, and terminal drought. And I would like to thank Crop Update to provide me with such an opportunity to come along and share the story with you. And Seth Rengel and Susan Barker are the co-author of this presentation or paper. I've been asked to provide three key messages, and I'm hoping to convey the three key messages through my short talk. The first message is ion toxicity in transiently waterlogged acid soil reduce root and shoot growth of intolerant wheat genotypes more in intolerant genotypes. The second message is drought accentuate the adverse effect of ion toxicity in root. It is a textbook knowledge that the drought accentuate adverse effect of other soil constraint, including deficiency or toxicity of other elements. But particularly, I would like to put an emphasis that drought also accentuate adverse effect of ion toxicity. And for those working on a drought, I would like to draw their attention that, it is a complex trait. Definitely, we need to have an extensive and tolerant root system to be able, to grow and dip into the soil profile and absorb water and nutrients. The third message is wheat genotypes with enhanced tolerance to ion toxicity produce greater yield than intolerant genotypes in acid soil prone to those constraints. And the explanation would be on the deep root system to allow uptake of water in acid soil with a drier surface layer is contingent on tolerance to ion toxicity. Ion toxicity are induced in transiently waterlogged acid soil. This is a wheat paddock at Kojonup, an acid soil site affected by waterlogging. As you might see the adverse effect of waterlogging in this paddock. Some area is severely affected by waterlogging, and climate change is going to reduce the quantity of the rainfall, but definitely it is coming along with intense rainfall early in the season, which is intensifying the waterlogging in most of the soil. If you walk along the gradients of waterlogging from one spot which is non-waterlogged and take the shoot and soil sample, you will notice that the concentration of these elements, manganese and iron, is very low. And if you walk to the extreme end, you will notice that the concentration of these elements is increasing several folds. A simple explanation for this phenomena is under reduced environment and lack of oxygen. Microorganisms are looking for alternative electron acceptor and reducing furthermore iron and manganese to the reduced form and increasing their concentration beyond the crop requirements. So it will become toxic to the plant. This is basically a brief graphic version of our first observation. We observed it long ago and published it. And that time it raised a question, whether we could, improve performance of the crop under waterlogged acid soil by enhancing tolerance to iron toxicity. I don't think I need to mention that the waterlogging is a complex phenomenon. It's the cause of many changes at the plant and soil level. And what I am concentrating here is iron toxicity and putting more emphasis on aluminum, manganese, and iron. Aluminum is well-established area, but manganese and iron are two other components of acid soil, ignored component of acid soil. And our predecessor couldn't, substantiate importance of these two elements, so they came along easily and say the acid soil is equal to aluminum tolerance or aluminum toxicity, which is quite wrong. At the time we raised that question, we had no access to required tool. What's the required tool? The required tool is near isogenic line for iron toxicity. At that time, we had no access to near isogenic line, so the alternative, option or substitute would be genotypes, extreme in their tolerance to each of these ions with similar low background to others. And to find the alternative, we looked at the Australian with germplasm, and we noticed the variation for tolerance to aluminum. It is, as I said, it is not surprised to have such a big variation for tolerance to aluminum toxicity within Australian germplasm, and it has been active selection pressure for aluminum, and those companies or breeding groups which have active selection pressure for aluminum, they have got, very well established, aluminum tolerance in their germplasm, and there are some companies which aluminum tolerance hasn't been a priority, so basically their genotypes or germplasm have got no tolerance to aluminum. And so it is a well-established area. I don't want to put an emphasis, but it is one of those, toxic ion, which is important under reduced environment, beside two other components, which are manganese and iron. In the case of manganese, we have got a very wide variation within Australian germplasm, and there are genotypes which are performing better than international indicator genotypes within Australian germplasm, and there are some which are quite intolerant to. So there is a wide variation, and considering the fact that we haven't got a selection pressure or for manganese toxicity, the simple explanation of these variations should be behind inactive or passive selection. Another potential for presence of such a big variation for manganese tolerance within Australian germplasm could be behind the co-tolerance of manganese toxicity and manganese deficiency. We have got the evidence that a couple of genotypes or wheat varieties which are tolerant, manganese efficient or tolerant to deficiency of manganese are also tolerant to manganese toxicity. And also the same is true about the iron, and we have got also a big variation within Australian germplasm for tolerance to iron. And here you could see some of the varieties are performing better than international indicator tolerant genotypes. This is Cetesaurus, which is tolerant to toxicity of iron, and this is one of those Australian entries which are performing more or less better than Cetesaurus. And there are some which are performing worse than, intolerant genotypes. So basically, this study, which took us, a couple of years, enabled us to optimize and develop some screening technique. At the end of the day, it enabled us to pick up genotypes which are extreme in their tolerance to iron toxicity, but with low level of tolerance to other. That enabled us to proceed with test of our hypothesis. The growth and grain yield of tolerant wheat genotype is less affected in waterlogged acid soil. We took up these 3 pair of genotypes with significant differences in tolerance from aluminum, manganese, and iron, and grow them in 3 to 4 acid soil a couple of times, and waterlog them, and stopped waterlogging and grow them up to maturity. As you can notice, waterlogging in acid soil reduces root growth of the genotypes, doesn't matter tolerant or intolerant genotypes, but most severely in intolerant genotypes. This is intolerant genotypes, this is a tolerant one. So you will notice that the root growth of the intolerant genotypes of aluminum, manganese, or iron is more severely affected under waterlogged acid soil. And when we grow them, and when we grow them up to maturity, you will notice that aluminum tolerant, manganese tolerant, and iron tolerant genotypes are performing better than intolerant genotypes. This is the pair. Another significant development is crop developmental stage of intolerant genotypes was affected by stress, and it is delaying the maturity of intolerant genotype by 7 to 10 days. And this is a serious problem of, besides, reducing the root growth of intolerant genotypes, the phenology or developmental stage of intolerant genotypes is delayed by 7 to 10 days. It is a serious problem where we are going to have a terminal drought. Terminal drought is affecting or reducing the performance yield of those intolerant genotypes worse than tolerant genotypes. Tolerance to iron toxicity enhances yield in drought-prone acid soil. Basically, as I mentioned already, drought is accentuating the adverse effect of all sorts of deficiency or toxicity on the root growth. This is a textbook knowledge. And we took the same genotypes and grew them in a few plus and minus lime stripes, which Blackley kindly shared with us at Wongan Hill and Meriden for 2 years. And we grow the same genotypes basically at Esperance Down Research Station. 2010 was an extremely dry year, extremely dry year. And we noticed that in Esperance, which the rainfall was, close to long-term average, all these genotypes showed a similar yield potential. Basically, there were no differences in expression of the yield of these genotypes. Basically, it means that these genotypes have got similar yield potential. But when we grow them in Meriden, in also Wongan Hill, 2010 was an exceptional opportunity to assess and evaluate performance of these genotypes under combined soil acidity and drought. And we noticed that aluminum tolerant and manganese tolerant genotype overperform intolerant genotypes in Meriden and Wongan Hill. So this is another addition to that tolerance of these genotypes under waterlogged condition, and also later on under drought. Basically, this is field experiment, 2 sites, which was under natural drought, but we have got, furthermore advanced tool to verify this, phenomena under rain shelter or at the meridian. So, hopefully, I've provided you with enough evidence that with genotypes with enhanced tolerance to individual ion toxicity produce greater yield. I've put a very conservative figure of 5 to 10%, very conservative, than intolerant genotypes in acid soil prone to transient waterlogging and terminal drought. What we are doing currently, we are assessing, evaluating variation within Australian advanced with varieties and advanced breeding lines, and also with varieties which is going to start quite pretty soon. And this work will provide with breeders with an opportunity to look at their genotypes or varieties or advanced line and to find the strength and weakness of the varieties. And also sharing this knowledge with grower will put them in a position to pick up the best variety which is more suitable to their paddock. But what would be the next step? The next step, we are in a position, to validate our finding using near isogenic line. And the more exciting step would be to test whether enhanced tolerance to combined ion toxicity could increase yield further in acid soil prone to waterlogging and drought. And we have got this tool, which is the result of 7 back crosses and pyramiding the tolerance. And so we have got the whole required tool to proceed with this next step, and also the required to go further to genetic and molecular study of the trait. And thank you.